Everyone, in this video I'm going to talk about making statistical comparisons to follow up on our lab exercises. So, first couple slides will be a refresher. Just going to talk a little bit about variables again, a little bit about graphing, and then I'll dive into the statistics. So as a reminder, when we're designing an experiment, we're going to structure experiments with two different kinds of variables, an independent variable and a dependent variable. And the independent variable is a variable that you're going to manipulate in the experiment, and it will wind up having two and sometimes more treatment groups. Often you'll have a control treatment and an experimental treatment. And that variable's something that you have expected to influence your dependent variable. And so then in turn your dependent variable is what you expect to change in response to your independent variable. And we don't actually know what it's going to be, so this is what we're actually measuring. When it comes time to make a graph, our convention for graphs of experimental data is to put the independent variable on the x-axis and the dependent variable on the y-axis. So here's a simple graph where we're comparing two different genotypes. The wild type genotype, which we probably consider to be a control treatment, and a mutant genotype called the rad9 mutant, so that would be our experimental treatment. And then on our y-axis, we have the percent that have survived after 30 seconds spent on a UV light box. So this is what we're measuring, what we think is influenced by the independent variable. Before we dive into statistics, we need to talk for a second about how to summarize a bunch of measurements that vary. So over here on this piece of paper, I've got just a set of numbers. You know, we can imagine there's some grades, right? And from just looking at these numbers, they vary. So the lowest is 2, the highest is 9. You know, there's some differences across here. And we use two different things to summarize measurements that vary. Those two things are some indicator of centrality and some indicator of spread. And this slide's a little busy, but we're going to walk through what those ideas mean. So to do that description, we're going to use some descriptive statistics, and we'll start out by summarizing some characteristic value of our data. So here's an example of actually 3 different measures of centrality, the mode, the median, and the mean. I'm not going to talk too much about the mode or the median. We're just going to focus on the mean at this point. So the mean is often also casually referred to as being the average. So for our mean, we'll just add together all of our numbers, divide by the number of numbers, and then we'll have a mean. So that's going to tell us, you know, what's a typical measurement out of this set of numbers. The second one is a little more complicated. That's a measure of spread. There's actually multiple measures of spread as well. So, you know, we can look at these numbers, for example, and say, well, they range from 2 to 9. That's one measure of spread. We're going to work with a slightly more sophisticated one called the standard deviation. This is just, again, definitions for those measures of centrality. So this is a bit wordy, but we'll get through it. So standard deviation, the definition of that is it's the average or standard difference of each value in a set from the mean. So again, you know, here's the set of grades from this class. We can calculate our average, add them all up, divide by the number of numbers, and we're going to get an overall average or mean of 5. And for the standard deviation, we'll start out by looking at the deviation or difference of each value from that average. So for number 2, that's going to be 2 minus 5. And then we take that quantity squared. That gives us 9. Here is going to be 4 minus 5 squared, so that's 1. Okay, so we take this square so that we don't wind up with negative values, because those could cause some problems. Okay, so continuing with our definition of the standard deviation, the first thing we wind up calculating is the mean of these squared values. And this is you don't need to know this term, but this is the variance. So if we add these up, divide by the number of numbers, we're going to get 4. as our variance. So again, it's a measurement of how different these are from our general pattern, from our mean. And then the standard deviation is just the square root of the variance. So if we take the square root of 4, we're going to get 2, and that is our standard deviation. So basically, the standard deviation, we squared everything here. We're just undoing that square, so we're back in the same measurement units as our initial measurements. So what we can think of is for our measurement of spread, we have a mean of 5. And most of our points are somewhere in the ballpark of plus or minus 2 around that mean. You'll notice that we do have some numbers that are a lot higher than that. So this standard deviation is just kind of, in general, where are all these measurements found. Great news is you don't have to do this calculation by hand. Excel will do it for you using the stdev function. All right, so that's our first step. So getting a very compact summary of the overall pattern for a collection of measurements. But the next thing we want to be able to do is, let's say we have 2 different treatment groups and we want to compare them to each other. Are the measurements from one treatment group similar to or different from the measurements for another treatment group? So for this, we're going to use a t-test, and a t-test is simply asking the question of, are 2 sets of measurements the same or different from each other? So here's another graph where we have our independent variable patient group on the x-axis. We've got treatments, a control treatment, and a group given an experimental drug. And on the y-axis, we've got the percent of patients that have survived after 5 years, our dependent variable. So did this drug have an effect on survival? All right, so t-test. So this is getting into the mechanics. You don't need to know all the specifics right now. It's just to help you understand the test a little better. So the formula for a t-test compares the variation between the means of 2 samples, as well as a variation in the standard deviations of the 2 samples. So I've got a big nasty looking equation here that just shows sort of how this is all put together into a single calculation. And again, we don't have to do this by hand for this class. Instead, a computer will calculate the t-test for you. So it has that built in and will just give you the major outcome from it, which is the probability value. So you'll hear lots about p-values in the future. In general, the main thing to know right now is we're going to work with a cutoff p-value of 0.05. So like the slide said, this means that there's a 5% chance that the difference in the means was due to chance alone, and a 95% chance that a real difference existed. So anytime you get a p-value greater than 5% or greater than 0.05, then there's not considered to be a true difference between your 2 treatment groups in science. Whereas if your p-value is less than 5, less than 0.05, that means the 2 groups are different from each other. It's a little easier to make sense of this with an example. So here's an example. Let's say we're comparing how many pollinator visits 2 different types of plants get. So New York asters compared to New England asters. So you crunch your numbers, you get a p-value of 0.3. That means that there is a 30% chance that any difference in the numbers you get for the means for these 2 aster types is due to chance alone. That's a pretty high potential for that outcome just as a result of chance, right? And so in other words, there's only a 70% chance that there was a real difference in the number of visitors between the 2 types of flowers. This is not a big enough margin to be considered a significant difference in science. And so here, the statistical term is we'd fail to reject our null statistical hypothesis. Now, it doesn't mean we failed, right? This is a specific use of the word fail. This means we can't reject our null hypothesis. when we're interpreting that biologically, that means our biological hypothesis has not been supported. There's no difference in the number of pollinator visits. So the aster type is not impacting pollinator visits. So just to reiterate this, if you get a p that is bigger than 0.05, you fail to reject the null statistical hypothesis. But if your p is less than or equal to 0.05, then you can reject the null hypothesis. So that's a situation where your biological hypothesis might be supported. So we're going to practice this. Here's a couple examples. So let's say we have the prediction that isopods prefer a moist environment relative to a more dry control environment. So our null statistical hypothesis would be increased moisture has no effect on isopod distribution. So if you get a p of 0.95, what would you conclude? Think about it for a second. So in this case, you would fail to reject the null statistical hypothesis because your p is bigger than 0.05, and that means that your biological hypothesis is not supported, right? So it seems that moisture doesn't impact where you find isopods. So here's another one. Heavy metal music increases the heart rate of listeners. So think for a second, what would your null statistical hypothesis, your null prediction be? Hope you came up with something like this. So heavy metal music does not affect heart rate. And now in this case, let's say you get a p value of 0.019. What should you conclude? I hope you can see you can reject the null statistical hypothesis, and this will support the biological hypothesis. So heavy metal music does affect heart rate. Here's sort of a graph that visualizes this, where you're in an area where you're pretty confident there's a real difference between your treatment groups. So p is less than 0.05, where in this other area, statistically we can't be confident we have a real difference. There's no statistically significant difference. So that's the whole concept of a t-test. What you'll need to do is see if, using a t-test, if the hypothesis we tested in lab, whether that is supported or not.